Welcome to Part 1 of Module 3 of the Foundation Certificate in Trauma-Informed Practice with Children and Families, an online certificate offered by the University of Vermont's Child Welfare Training Partnership. This module covers the topic of child and adolescent brain development and behavior based on course material developed by Dr. Jim Hudziak, MD, and Dr. Matt Albaugh, PhD, at the Vermont Center for Children, Youth, and Families. Upon successful completion of this course, participants should possess a greater understanding of the human brain, including the general function of brain regions, and be able to describe some of the developmental processes that serve to shape the child and adolescent brain. So as we get started, we will first spend some time getting to know our own brains a little better. We will begin this journey by starting at the cellular level and examine some of the basic building blocks of the human brain. This terminology will help us better understand some of the concepts introduced later. It is important to note that this is just a general overview and in no way is the following a comprehensive primer on neurobiology. We will just be covering the basics. There are approximately 100 billion neurons in the human brain. Generally speaking, neurons are comprised of dendrites, which are the branch-like extensions of the cell body making connections with other neurons. Their primary function is to connect information from other neurons. The cell body, or soma, which includes the nucleus, its primary function is to integrate electrical signals. Axon, a single cylindrical structure, the primary purpose is to conduct information to other neurons. And axon terminals. The primary function is to transmit information to other neurons. Electrical signal flows in one direction, from dendrites, through cell body, down axon, to axon terminals. Information does not flow backwards. So how do neurons communicate with one another? Neurons connect with other neurons at locations called synapses. When an electrical signal propagates down the axon to the axon terminals, which are filled with vesicles full of neurotransmitters, neurotransmitters are released into the synapse and travel across the synaptic cleft to the postsynaptic neuron. Depending on the neurotransmitters and receptors involved, this affects the functioning of the postsynaptic neuron. So now we are going to zoom out from the cellular level and take a more macroscopic view of the human brain. At a very gross level, the brain consists of white and gray matter. Gray matter is comprised of neuronal cell bodies. For all intents and purposes, we can think of the gray matter as where the brain's computations are taking place. White matter is mainly comprised of myelinated axons. We can think of white matter as the brain's wiring, transmitting information to other gray matter regions. One gray matter structure that we will be focusing on is the cerebral cortex. This is the most evolutionary recent gray matter structure. The cortex is a sheet-like structure making up much of the brain's outer surface and is full of gyri and sulci, essentially folds that allow for maximizing surface area. The cerebral cortex makes many connections with gray matter structures deep in the brain, subcortical gray matter structures like the thalamus and basal ganglia, as well as other parts of the cortex. Although we might be tempted to think of the cortex as a relatively small outer layer of the brain, it astoundingly accounts for roughly 70 to 75% of the brain's volume. Next, we will be talking about the lobes of the brain. The lobes are named after the portions of the cranium that sit atop them. The brain is comprised of two hemispheres, with 4 lobes in each hemisphere: frontal, parietal, occipital, and temporal. The occipital lobe houses the brain's primary visual cortices. Damages to the occipital lobe can affect our perception of movement and color. The temporal lobe has no single function and is intimately involved in the processing of auditory and visual information, particularly object recognition. It also possesses medial temporal cortices that are critical for memory formation, more specifically, long-term memory formation. The temporal lobe also houses brain areas that are central to emotional behavior and the processing of emotional information. The parietal lobe is involved in visuospatial processing and is directly involved in integrating across sensory modalities. The parietal lobe shares robust anatomical connections with the frontal lobes, including frontal motor areas, helping to integrate information and guide movement. Damage to the parietal cortex, particular to the inferior parietal lobe, can markedly impair how we perceive the world. Damage to the inferior parietal lobe can cause hemispatial neglect, a condition that is characterized by a profound deficit in awareness of one side of space, typically the side contralateral to the lesion. As we will discuss at length, the frontal lobes are involved in many different processes, including planning and organization, movement and fine motor coordination, working memory, inhibitory control, we can think of this as akin to the brakes in an automobile, as well as the ability to regulate emotions. As we will see, this part of the brain has the most protracted developmental trajectory, and portion of the frontal lobes continue to mature well into young adulthood. So now, we will move on from the cerebral cortex. There are a number of subcortical gray matter structures in the human brain. In general, we have the thalamus, basal ganglia, which include a number of areas such as the caudate nucleus, putamen, and pallidum, and limbic structures like the amygdala and hippocampus. All of these structures share robust anatomical connections with the cerebral cortex. Please note that the term striatum can be used to refer to the combination of caudate nucleus, putamen, and ventral striatum. The name striatum refers to the striations or lines that can be seen between the caudate and putamen in axial and coronal cross-sections. These striations are actually projections. The striatums share close anatomical connections with the cerebral cortex. The ventral striatum is also known as the nucleus accumbens, NA, and constitutes a central node in the brain's reward pathway. The NA has very close connections with the ventral and medial portions of the prefrontal cortex. Dysfunction within these circuits has been tied to addiction and substance use behaviors. There are classes of circuits that involve parts of the prefrontal cortex, the caudate, and the putamen, and the thalamus. Although it is beyond the scope of today's training, these circuits are referred to as cortico-striatal, thalamic, cortical circuits, and they play an important role in executive functioning and working memory. Limbic regions, including the amygdala and hippocampi, have long been implicated in the mediation of emotional processes, including aspects of fear and anxiety. The amygdala, in particular, are central to the neural circuits that have evolved to detect potential threats, as well as respond to salient stimuli in the environment in youth and adults. In both human and non-human primate imaging studies, heightened amygdala reactivity has been associated with dysregulated emotional behavior and anxious depressed symptoms. The advent of diffusion tensor imaging, DTI, a type of MRI, allows for in vivo mapping of white matter pathways in the brain. In this slide, major white matter pathways are shown in lateral view. This slide shows white matter pathways in dorsal view. The fiber pathways of the brain are shown here in anterior view. The corpus callosum is the pathway that connects the right and left hemispheres of the brain to communicate about important functions such as arousal, emotion, and cognition. The superior longitudinal fasciculus relays messages related to executive functioning. The corticospinal tract relates to movement and motor function, and the cingulum bundle transmits information about emotional behavior. This concludes our basic primer of human brain anatomy. We will now move on to talking about early brain development. In line with the training aims of this module, the following is not meant to be a comprehensive overview of brain development. The following provides a general understanding of some of the processes taking place beginning in utero. The central nervous system remains sensitive to teratogens for nearly the entire window of prenatal development. The brain is vulnerable due to rapid changes that are taking place. Early brain development involves a flurry of changes. We will spend time discussing the following: neurogenesis and the migration of cells, Nerve cells form by the division in the ventricular lining of the brain. In the ventricular lining, each cell gives rise to two daughter cells that either migrate or undergo further division. Migration of the cells to various brain areas may continue postnatally. The exact timing of migration depends on the brain area. The mechanisms that control migration are still not well understood. Cells migrate away from the ventricular zone and eventually reach the surface of the brain. Upon doing so, the formation of axons and dendrites starts. As new cells arrive, they travel through the layers that are already set up, and as a result, form a new outer layer. Formation and growth of axons. As soon as neurons reach their destination, they start developing axons. Axons grow at a rate of 7 to 170 micrometers per hour. The branching of axon occurs at the growth cone. Incredibly, axons possess specific targets that must be reached if the neuron is to become functional. If axonal growth is disrupted, for example by lack of oxygen or particular tetrogens, there can be devastating effects. Formation growth of dendrites. The growth of dendrites parallels that of axons. The growth of dendrites, amazingly, is timed to intercept axons that are supposed to innervate them. Dendrites also possess growth cones. The growth and formation of dendritic processes continues postnatally. Synaptogenesis. With the growth of axons and dendrites, the number of synapses also increases. In humans, synaptic density is believed to increase up to roughly 2 to 4 years of age, after which about 50% of synapses are lost by age 16. As is shown in this slide, the exact timing of peak synaptic density depends on the brain area. Myelination. As we discussed earlier, myelination refers to the glial cells insulating axons, thereby increasing the speed and efficiency of signal transmission. Myelination has also been tied to aspects of cognitive development. And now we move into postnatal brain development. The human brain undergoes rapid development during the first few years of life. At birth, a baby's brain contains 100 billion neurons, approximately equal to the number of stars in the Milky Way. At birth, the average number of synapses per neuron is 2,500, but by age 2 to 3, it's about 15,000 per neuron, 5 times the connection. By the age of 3, a baby's brain has reached almost 90% of its adult size. The development of synapses occurs at a very rapid rate during the first couple years of life. At age 2 to 3, the brain has up to twice as many synapses as it will have in adulthood. Estimates suggest that during this age range, children have roughly 1,000 trillion synapses. These extra connections are gradually eliminated during childhood and adolescence in a process referred to as pruning. The metaphor of a bonsai tree can be used to explain pruning. For most of development, the interplay of genetic and environmental influences serves to prune these connections, with connections that are used the most surviving, and connections that are not utilized are sheared away. This affords a great degree of adaptation. However, as we will see later in part 2 of this module, when a child's brain is adapting to an adverse environment, this can result in behaviors, viewed in certain contexts, as maladaptive, for example, aggression and anxiety. Now, we will move on to learn about cortical development. MRI methods exist allowing researchers to accurately measure the thickness of the cerebral cortex at a very large number of points. Cortical thickness is defined as the shortest distance between the outermost surface of the cortex and the interface between cortical gray matter and white matter. Measuring cortical thickness can be done repeatedly during the course of development, and researchers can measure age-related changes in thickness. Most longitudinal studies of cortical development suggest that, very early on in development, there is an increase in cortical thickness. The exact age of the peak depends on the individual and cortical area, followed by thinning that continues into adulthood. It is postulated that these quadratic or inverse U-shaped thickness trajectories represent an early increase in connections, paralleled by increases in cortical thickness, followed by subsequent pruning and elimination of unused connection, paralleled by cortical thinning. Note that the posterior portion of the brain, comprised of occipital and parietal cortices, are maturing earlier relative to anterior frontal portions of the brain. Some of the most breathtaking visualizations with regard to cortical development come from a landmark study by Dr. Nitin Gagte and Jay Giad in 2004. A series of slides are included showing various stages of development from 4 to 21 years of age. Age 4. Note early maturation of visual and motor sensory cortices and large expanses of red, thicker, less mature prefrontal areas. At age 9, aspects of the superior parietal lobes are becoming more mature, which likely ties to aspects of visual spatial ability in mathematics, as well as visually guided movement. It is interesting to note that at age 13, the capacity for creating emotions increases at age 13, but the development of prefrontal cortex, which is responsible for decision-making, self-control, judging risk, and making long-term plans, lags behind. This help explains why adolescence can seem emotionally out of control. The maturing prefrontal cortex of a 17-year-old is capable of abstract thought, planning, risk-taking, and decision-making. Even at 21, portions of the prefrontal cortices appear relatively less mature when compared to other areas. These portions continue to develop among young adults in their 20s. Just as the cerebral cortex undergoes remarkable changes, so does the white matter of the brain. In general, studies investigating white matter development across childhood and adolescence have reported widespread fractional anisotropy (FA) increases in white matter pathways, reflecting fiber density, axonal diameter, and myelination in white matter. Such age-related FA increases in cerebral white matter are postulated to reflect increasing microstructural integrity and presume functional maturation of cerebral fiber pathways. Indeed, age-related FA increases within specific white matter pathways have been found to be uniquely associated with aspects of cognitive performance among typically developing 6 to 17-year-olds. When looking at the different developmental trajectories of various white matter pathways, note the peak in structural integrity is later in life, similar to what we saw with cortical development. For example, the superior longitudinal fasciculus (SLF) is a large white matter pathway connecting frontal and parietal cortices and has been implicated in aspects of executive functioning and attentional skills. Note that its maturational peak is in the 20s. Given these very dramatic changes in cortical and white matter structure, it is probably unsurprising that, during the same developmental window, we also see dramatic increases in cognitive abilities. These data are courtesy of the NIH Normal Brain Development Study. We see an exponential increase reading comprehension between the ages of 0 to 200 months, or 0 to 18 years. Verbal IQ, measured using the vocabulary subscale on the WASI, also dramatically increases during this developmental window. Lastly, we see a sharp age-related decline in working memory errors between the ages of 0 and 18. Indeed, in a study by Shaw and colleagues, 2006, cortical thickness trajectories in superior frontal areas were linked to different levels of cognitive ability. Interestingly, superior intelligence was associated with the most accentuated thickness trajectories. Let's take a closer look at adolescence and the brain changes that characterize this developmental stage. There is no definitive age range that defines adolescence. For our purposes today, we are referring to 12 to 17 years of age. For this activity, please answer the following questions. What do you believe are the defining features of this age range? What are the challenges? Are there behaviors that typify this age range? Based on what we have covered, do people have some guesses as to what aspects of brain development are associated with behavioral changes during this time? There are many positive changes that are taking place from childhood to adolescence, including increased strength, speed, reaction time. As discussed, we also see improvements in mental reasoning and cognitive ability. We also see enhanced survivability, with increased resistance to environmental stressors like extreme temperatures, hunger, etc. Despite these gains in strength, overall morbidity and mortality rates increase from 200 to 300% from childhood to late adolescence, especially among males. Adolescence is often characterized by heightened emotionality. We probably can all remember the intensity with which we experience emotions during our teenage years. During this developmental window, there is also a rise in risk-taking behaviors and substance use initiation. We also begin to see the formation of close interpersonal ties to peers, individuals outside of the family. We also have a flurry of hormonal changes that are taking place as part of puberty. Given the density of sex hormone receptors in the brain, particularly in limbic regions of the brain that are central to emotional processing, areas of the amygdala and hippocampus, puberty can have dramatic influences on brain structure and function. There is also an increased drive for independence and autonomy, which, oftentimes, can manifest in conflicts with caregivers. So, from a brain development standpoint, why is adolescence often a tumultuous time for individuals? Let's take a closer look. As we have already touched upon, there are developmental processes that are still shaping the brain during adolescence. One process is myelination. Myelin serves to insulate axons and speeds the relay of electrical impulses within the brain, affecting cognition, decision-making, impulse control, and emotional regulation. Another process is synaptic refinement, particularly at the level of the cortex. Remember back to earlier when we discussed that from age 2 to 3 to late adolescence, the number of synapses is reduced by 50%. This elimination process is shaped in part by a young person's environment and experiences. Remember that aspects of the prefrontal cortex haven't matured until well into young adulthood. This brings us to what has been coined the developmental mismatch hypothesis. This hypothesis has been largely advanced by Dr. B.J. Casey and her colleagues at Cornell Medical School. There is evidence from both structural and functional neuroimaging that regions such as amygdala and ventral striatum nucleus accumbens matures more rapidly relative to portions of the prefrontal cortex. Given what we already know about the amygdala from an evolutionary standpoint, why might it make sense for more regions like the amygdala to mature more quickly? Given that this is an important structure for fear, and fear is a critical emotion for survival, it might make sense from an evolutionary standpoint for the amygdala to mature faster. Similarly, it might make more sense for reward areas to mature more rapidly relative to prefrontal areas that are important for assessing long-term consequences of actions, other people's perspectives, higher order cognitive skills, etc. The figure in the top portion of the slide illustrates this mismatch in developmental trajectories. The top curved line represents the developmental trajectory of a subcortical region like the amygdala and nucleus accumbens. Notice that this line has a steeper slope at earlier ages, indicating more rapid maturation. Conversely, the bottom line represents the developmental trajectory of the prefrontal cortex. Note that this trajectory is less curved and more linear, representing more protracted maturation. It is postulated that there is greatest risk vulnerability during the developmental window, where this is the most space, shaded in figure, between these two lines. Now, let's talk a little bit more about how limbic structures like the amygdala interact with portions of the prefrontal cortex, and why these interactions are central to emotion regulation. Research suggests that the interactions between limbic regions of our brain and the prefrontal cortex have a tremendous impact on how we think, feel, and behave. As we will discuss later, it is the development of the frontolimbic circuitry that is postulated to be central to the emergence of certain forms of psychopathology. Frontolimbic circuitry is also believed to be affected by adversity and trauma. Indeed, there is much evidence to suggest that parts of the prefrontal cortex, the amygdalae and the connections, i.e. axonal projections, between these brain areas are central to emotional regulation. Based on research findings, as outlined in seminal work by Phillips, Drevitz, and their colleagues in 2003, there are two general countervailing systems in the brain. One, a dorsal system that is anchored in dorsal cortical areas such as the dorsolateral prefrontal cortex, DLPFC, and parietal cortices. And two, a ventral system that is centered among the amygdala and ventral striatum. These two systems are reciprocally related, such as that one gets active, the other becomes less active. The dorsal system, shown in blue in slides, has been linked to aspects of cognitive emotion regulation, perspective taking, and problem solving. Conversely, components of the ventral system, the red portion, have been implicated in fearful behavior, fight or flight response, panic symptoms, etc. We already know this at an intuitive level. How well can we problem solve when we are emotionally dysregulated? This is particularly true in children for whom the dorsally situated network is still very much developing and maturing. Emotional self-regulation is achieved when we are able to use the dorsal system to think and engage in perspective taking and problem solving. Results from a study by Deborah Yergelong-Todd are displayed here. Because the prefrontal cortex is one of the last areas of the brain to mature, adolescents tend to use other areas of the brain, in this case emotional areas, when making decisions. For example, brain activity, seen here via functional MRI, shows that when judging emotion represented on a face, a teenager's amygdala is activated, on the right. Activity in the amygdala during this task likely reflects more of a gut reaction than a reasoned one. In contrast, the prefrontal cortex, involved in reasoning and reflection, is activated in the adult brain, shown on the left. The developmental mismatch that we have been talking about is believed to play a role in why, at least for some adolescents, this developmental window is a period of vulnerability. Looking at epidemiological data, overall rates of clinical depression explode during this age range. This is particularly true for females. Although there are certainly other factors that likely contribute to this prevalence data, it is believed that brain changes during adolescence may play an important role. If we go back to mortality data that we mentioned earlier, what types of behaviors do we think contribute to the sharp uptick in mortality rate during adolescence? There is a dramatic increase in novelty and sensation-seeking behaviors at puberty. Early maturation of the ventral striatum and the protracted maturation of prefrontal cortices are posited to underpin this phenomenon. The dual contributors to adolescent risk-taking, the shift in reward sensitivity that drives adolescents to seek higher levels of novelty and stimulation than they did as children, and the relatively slow maturation of the regulatory competencies that would rein in this novelty and stimulation seeking, do not operate simultaneously, and therein lies the problem. The shift in reward sensitivity occurs relatively early in adolescence, is mainly subserved by changes in the limbic system, and is probably closely tied to the neuroendocrinological changes in puberty. The maturation of self-regulatory competence is ongoing even in late adolescence, is subserved primarily by changes in the prefrontal cortex, and does not seem to be closely linked to pubertal maturation. It is better predicted by chronological age and years of education. The temporal gap between these two changes creates a special window of vulnerability in adolescence, vulnerability not only to risk-taking, but to other forms of psychopathology. This is also believed to play a role in the sharp spike in substance use initiation. The age group that is associated with highest rates of getting drug dependent for the first time is youth, before the age of 25. Survey data from a national study in the U.S. provides further supporting evidence. The findings indicate that there is a general steady increase of the rate of an alcohol use disorder from age 12 to 18. Then, the rate drops rather noticeably at age 19 and stays relatively lower throughout the remaining years into young adulthood. The results support the view that youth, particularly during the teenage years, is a high-risk period for developing an alcohol use disorder. To summarize, in Part 1 of this module, we learned about the dramatic changes that occur in the human brain from conception through adolescence and how these changes relate to developmental functioning in numerous domains. It is important to understand the complexity of normal human brain development in order to truly grasp the impact of adversity, including trauma, on children during critical periods of their development. Part 2 of this module will cover how adversity, including maternal stress and substance abuse in utero, and child maltreatment during childhood, can impact brain development. Part 2 will relate the impact of trauma on the brain to developmental and behavioral challenges frequently observed among children exposed to trauma. We will also explore resilience factors that can have a positive impact on brain development. Thank you for completing Part 1.